Hear the word mutation, and you likely picture glowing green ooze, comic book monsters, or catastrophic radiation. The biological reality is far more mundane. In the time it takes you to watch this video, thousands of mutations are quietly happening inside your own body. You aren't sprouting extra limbs or developing superpowers. The vast majority of these spontaneous internal changes occur without you ever noticing, completely harmless to your biology. At its core, a genetic mutation is a physical typo. It is a simple change in the specific sequence of bases along a DNA molecule. This long biopolymer acts as your master blueprint. Those sequences of bases spell out the exact genetic information required to build and maintain you. If DNA provides the precise instructions for life, we have to ask, what happens when the text in that instruction manual is accidentally rewritten? Before a single cell can divide, it has to copy its entire DNA sequence so that both resulting cells inherit the exact same code. The biological machinery that handles this massive copying job is highly efficient, but it isn't perfect. It regularly makes internal replication errors. Sometimes these errors are driven by the DNA itself. Certain sequences can temporarily fold into highly complex, unstable physical shapes. This table details the properties of common DNA forms. Looking at the zDNA column on the right, it forms a complex left-handed helix. These sequences have the potential to enhance the frequencies of recombination and deletion events. Outside factors force changes too. Triggers like high-energy radiation, chemical mutagens, and known carcinogens can physically damage the DNA strand. Regardless of whether the cause is a structural anomaly inside the cell or chemical damage from the outside, the result is the same. The genetic code is permanently miswritten. When we zoom in on the sequence, these typos generally fall into a few mechanical categories. Point mutations swap a single base for a different one. Insertions squeeze entirely new base pairs into the existing sequence. Deletions drop chunks of the code entirely, leaving the remaining sequence to snap together across the gap. Portions of this DNA specify single functional units called genes. Since genes dictate the instructions for building proteins, altering the base sequence alters the resulting proteins. Proteins do the heavy lifting of cellular function. Change their structure, and you change the physical traits, the phenotype of the organism. These alterations to an organism's phenotype produce three possible outcomes. First are the harmful outcomes. About half of the mutations that hit the protein coding regions of a gene are deleterious, potentially breaching the function and leading to disease or non-viable embryos. This chart shows the distribution of mutation effects across the genome. As you can see, the vast majority of mutations land in non-coding regions and are completely neutral, having zero effect on the organism's fitness. Then there is the beneficial category. A microscopic percentage of these genetic typos actually confer a functional fitness advantage. For instance, a non-lethal regulatory mutation in human Hox genes can actually increase the number of digits on a hand or foot, a condition known as polydactyly. If that organism happens to live in an environment where extra digits improve its ability to grip or climb, that novel trait provides a distinct survival advantage. When beneficial mutations result in higher fitness, natural selection steps in to favor the new phenotype. Individuals with the advantageous trait survive longer and reproduce more. Over generations, the frequency of that mutated allele replaces the ancestral version, spreading throughout the population. Because of this process, mutations are the ultimate source of all genetic variation in living organisms.